Hello students, welcome back to a new video of Sunanda's tutorial. In this video I will discuss some important points regarding your chapter plant growth and development. Please listen to all these points carefully because all these points are very important for your upcoming SSP, TGT, PGT and lectureship examination. It is also important for NEET 2025 aspirants. So all of you listen all these points carefully. So let's start. First point is plant growth may be defined as a permanent, irreversible change in size of a cell, organ or whole organism, usually accompanied by increase in dry weight. Simply weight. Second point, there are three phases of growth. One is cell division, where cell divide, then cell enlargement in the size, then cell mature. Maximum elongation occurs immediately behind the apex, apical part, and behind the apex zone of elongation is present. The time course of growth of an entire plant or plant organ plotted on a graph shows S-shaped or sigmoid curve. So the plant growth curve is a sigmoid curve. Growth in plant can be measured in terms of increase in length or girth or diameter, increase in fresh weight and dry weight, then increase in area or volume, increase in the number of cells produced. Then what are the methods of the measurements of the growth? First one is direct method, when we directly measure the length of the plant. Horizontal microscope, by using horizontal microscope, by using arc indicator or Pfeffer's oxanometer. In addition to nutritional and genetic factor, growth and differentiation of plants depend on some more important hormonal factors which regulate the distribution and fate of nutrients in different organs and the final form of plant body. The growth hormones are defined as organic substances which are synthesized in very minute quantities in one part of the plant body and transported to another part where they influence specific physiological processes. A group of plant hormones including auxin, gibberellins, cytokinins, ethylene, abscisic acid are presently known to regulate the growth. There are several synthetic chemicals which often resemble the hormones in physiological action. They are called growth regulators or synthetic hormones. Auxins include all those chemical substances which promote growth of the stem or coleoptile sections, and decapitated or apex removed coleoptile but in same concentration are incapable of causing growth of the intact plant. The principal naturally occurring auxin is indole 3-acetic acid IAA. Auxins cause cell elongation in the stem, cell division in the cambium, cell division in tissue culture, root initiation, apical dominance, parthenocarpy, enzymatic activity, prevent the formation of abscission layer. Then gibberellin are plant growth hormones which enhance the longitudinal growth of the stem when applied to the intact plant. The gibberellins, however, do not enhance the growth of isolated plant parts. Some of the important physiological effects of gibberellins are elongation of stem, reversal of dwarfism, bolting. Bolting means the elongation of internode, substitution of cold treatment or vernalization, cell division in cambium, parthenocarpy, helps in seed germination, helps in the breaking of dormancy and de novo synthesis of hydrolytic enzymes. Then cytokinins, these are plant growth hormones which act primarily on cell division and have a little or no effect on extension growth. Some of physiological roles of cytokinins are cell division, cell enlargement, initiation of interfascicular cambium, counteraction of apical dominance, breaking of dormancy, delay of senescence, accumulation and translocation of solutes. Ethylene is a gaseous hormone involved mainly in ripening of fruits. Abscisic acid is an inhibitor hormone, otherwise known as stress hormone, responsible mainly for induction of dormancy, inhibition of growth, and causes stomatal closure. Instead of acting separately, the plant hormones generally interact with each other in causing various growth and morphogenic effects. Development is a series of changes which an organism goes through during its life cycle. It is usually always associated with morphogenesis as well as differentiation. There are three distinct phases of plant development: phase of seed germination and vegetative growth, phase of reproduction, flowering, then senescence, and finally death. Requirement of specific day length for flowering is called photoperiodism, and the requirement of specific temperature, low temperature actually, is called vernalization for flowering. The role of photoperiodism in control of flowering was clearly demonstrated for the first time in 1920 by Garner and Allard. And according to the photoperiodic response, they categorize plant into short day plant, long day plant, and day neutral plant. Short day plant which requires shorter day length than the critical photoperiod or critical day length. And example: tobacco, soybean, cosmos, sugarcane, cockleber, and long day plant which require longer photoperiod than critical day length. Then day neutral plant, they are independent of any specific photoperiod. Photomorphogenic process such as seed germination, photoperiodism are controlled by photoreversible pigment phytochrome. And what is vernalization? Vernalization is the requirement of low temperature or chilling treatment for induction of flowering or for inducing the flowering. Okay, so these are some important points on the chapter plant growth and development. Thank you.